So, okay. All right. Thanks everybody for coming. I'm really excited about today's talk. We're going to be hearing about the evolution and biogeochemical impacts of bioturbation. So Lidya Tarhan is an assistant professor of Earth and Planetary Sciences at Yale. She did her master's and PhD at UC Riverside in geological sciences and has been working for a while on the evolutionary history of bioturbation. So from there, she did a postdoc at Yale and then stayed on as a faculty member. And I don't want to be too long, so we'll go ahead and get started, Lidya.
All right. Well, thank you, Kelly, for that very nice introduction. And I'm very excited to have the opportunity today to talk about some of my group's recent work on the evolutionary history of bioturbation. So, of course, as many of you are already very profoundly aware, bioturbating or burrowing and sediment mixing animals are some of our foremost examples today of ecosystem engineers, of organisms that shape their surroundings not only transiently, but also, as I'll be focusing on today, over timescales that are ecologically and geologically meaningful. And if we look at the seafloor today, we see abundant evidence for the many ways that bioturbators shape the chemistry, the physical nature, and the ecology of the seafloor. By mixing sediments, they oxidize them, enhancing rates of remineralization, stimulating bacterial growth, influencing the transfer of nutrients across that sediment-seawater interface, and by doing so, impact the biogeochemistry of not only the seafloor, but also overlying ocean waters. And by mixing and churning seafloor sediments, bioturbators increase their fluid content and porosity, transforming the upper, on average, 10 centimeters or so of the seafloor into a texturally massive homogeneous zone that we call the sedimentary mix layer, which you can see a very nice example from a modern box core. And this is from Richard Bromley's excellent 1996 book on trace fossils. And due in part to this potent combination of rheological and biogeochemical effects, bioturbators in turn strongly influence the structure of seafloor communities. And one of the questions that my group is very interested in is, if we look back in the geologic past, do we see similar evidence for bioturbators serving as critical ecosystem engineers? To what extent did ancient bioturbators shape the biogeochemistry of surrounding ocean waters or influence their productivity? And can we recognize correspondence between major changes in the geobiological records of the geologic archives and inflections in intensity or extents of bioturbation? And our predicted responses to some of these questions fall broadly into what we can think of as a couple of different camps. The first of these is a framework that's informed more closely by biogeochemical modeling, in which a number of recent studies have suggested that the emergence of bioturbation would have actually had a deleterious impact upon both productivity and oxygenation of ocean waters. And on the other hand, we have the predictions of macroecological and macroevolutionary theory that the emergence of bioturbation, like other forms of ecosystem engineering, should, on the other hand, enhance the flow and transfer of nutrients through ecosystems, thus sustaining greater organism size and greater ecological complexity. And I find this apparent dichotomy quite fascinating. Does this mean that one of these frameworks must be fundamentally flawed? Or could there potentially be some way to reconcile them? But before we can really dig into some of the impacts of ancient bioturbation, we need more robust first-order records of how bioturbation has changed over our planet's history. So if, for instance, we look to the geologic past, when do we first start to see evidence in stratigraphic archives of appreciable sediment mixing, of the development of a true sedimentary mix layer? And when do we first start to see evidence that seafloor sediments were becoming mixed to the extents, to the intensities and depths that they are mixed on the seafloor today? And this is a critical question because historically, the advent of bioturbation, as approximated from the trace fossil record of fossilized trackways and trails and burrows, such as this burrow, Treptichnus pedum, which is actually our index fossil, our marker in time for the Precambrian Phanerozoic transition, 540 million years ago, and one of the most important boundaries in the history of life. So on the basis of this trace fossil record, it's been widely suggested that bioturbation was responsible for a pretty rich range of geobiological and paleontological phenomena that we observe in the geologic record, such as declines in the abundance and diversity of microbial buildups of features like stromatolites, the disappearance of some of the earliest complex animal-like life forms, like the Ediacara, closures of windows of exceptional fossilization, the rise of oxidants in the ocean, and even the stabilization of Earth's long-term carbon cycle have all been blamed on the advent of intensive sediment mixing. But were earliest Phanerozoic seafloor sediments as well mixed, as well bioturbated, as they've commonly been assumed to have been on the basis of this trace fossil record? And to what extent do records of geobiological change actually align with major inflections in either the intensity or the extent of bioturbation? And historically, this has been something of a thorny question to unravel, in part because there's been a heavy reliance upon those trace fossil archives, particularly trace fossil specimens archived to museum collections, as well as trace fossil assemblages described in the ichnological literature. And although trace fossils can be enormously informative in telling us about the nature and the timing of major biological innovations, they don't necessarily give us a representative snapshot of background conditions on the ancient seafloor. And in particular, trace fossil specimens archived to museum collections are commonly skewed towards some of the largest and the best preserved examples, and they're commonly reposited without much sedimentological context. Moreover, a number of the studies that have argued for an early escalation in intensities of sediment mixing with saturation at modern-like values of bioturbation intensities and depths, even as early as stage 2 of the Cambrian, so a mere 10 million years after the Precambrian Phanerozoic transition, have tended to rely heavily upon maximum metrics of bioturbation, like maximum depths of burrowing and maximum intensities of sediment disruption. However, maximum metrics for bioturbation won't necessarily give us a representative understanding of typical levels of bioturbation on the ancient seafloor. And as demonstration of this, we can look to particularly well-characterized Cambrian and Ordovician, so spanning about 540 to 445 million years in age, carbonate-dominated successions from the western US, which record a variety of shallow marine depositional environments, and which have been extensively characterized from a bioturbation perspective. So what I'm showing you here are 6 plots representing 6 different geologic formations from this Cambrian and Ordovician data set. And these are representative examples of a much larger data set that spans collectively thousands of meters of stratigraphic section over which bioturbation data have been continually logged. And along the x-axis of each of these plots are 3 older examples of Cambrian age. On the top row and our 3 lower examples of Ordovician age, we can see a very commonly used metric for bioturbation intensity. This may be familiar to some of you, but for any for whom it's less familiar, this is the ichnofabric index. And this is a semi-quantitative metric for the extent to which sedimentary depositional fabrics, so physical stratification with which sediments are deposited along the sea floor, is disrupted or overprinted by bioturbation. So if we were to look at a sedimentary column or geologic succession, edge on and cross section, an ichnofabric index value of 1 corresponds to no disruption, just that physical stratification, so planar lamination, cross bedding. An ichnofabric index value of 5 means intensively disrupted to the extent that there's no longer any vestige of that physical stratification. We only see overprinting burrows. And an ichnofabric index value of 6 is bioturbation so intensive that it's essentially self-erasing, that not only do we have no vestige of the original depositional fabric with which those sediments were deposited, we no longer see discrete burrows either. We simply have a texturally massive homogeneous fabric, so essentially a true sedimentary mixed layer. So what we can see across these 6 successions, which range from about 50 meters to 150 meters in thickness, is that we do see some strata that are characterized by very high intensities of bioturbation, of ichnofabric index values of 5. However, the relative abundance of those more intensively bioturbated strata is actually quite low. And we see that the majority of each of these units is comprised of things lower on that scale of poorly to moderately bioturbated strata. So clearly, this is an instance in which maximum metrics for bioturbation intensity are not representative of extensive bioturbation throughout the majority of these shallow marine carbonate successions. And the extent of variability that we see in these Cambrian and Ordovician examples really shouldn't surprise us when we consider the extent to which bioturbation varies on the modern seafloor. So here you're looking at a global heat map of the depth of the sedimentary mixed layer for the modern oceans. And this was built by myself and colleagues from all currently available empirical sedimentary mixed layer data. This is drawing on Martin Solin's recent compilation published a few years ago in Scientific Data. And in addition to those empirical records, what we've done is we've used a machine learning framework, and specifically a random forest tree-based ensemble, to predict at a much higher spatial resolution than we have access to only from those empirical records, the distribution of different depths of the sedimentary mixed layer using a range of ecological and environmental predictor variables. So we can see that there's quite a lot of heterogeneity in the depth of the sedimentary mixed layer even today, but to take a maximum ichnofabric index from one of these ancient successions and suggest that that's representative of the entire global ocean at that time would be akin to my highlighting one of these bright red areas, for instance, in the equatorial Pacific, and suggesting that that's representative of the entire oceans today. But before we can really dig into what either the biogeochemical or the paleoecological consequences of ancient bioturbation may have been, we need better first-order records constraining how background levels, different extents and intensities, and also different styles of bioturbation varied through time. In particular, I think there's a pressing need for such records spanning the early portions of the Phanerozoic eon, the Cambrian and Ordovician and Silurian periods that encompass the first 120 million years of the Phanerozoic and witnessed a number of major radiations in animal life, such as the Cambrian Explosion and the Great Ordovician Biodiversification Event, and also saw the establishment of many of the major ecologies that characterize animal communities living on the seafloor today. So if we look at successions of this age, it's readily apparent from the trace fossil record that animals began to colonize the seafloor quite early in this interval. As early as the earliest Cambrian or even the very latest Precambrian, we see a variety of different burrow architectures, including vertical burrows, recording animals exploiting sediments in a more three-dimensional manner. But what about the more sedimentological side of bioturbation? To what extent and what intensity were these early Paleozoic sediments well mixed? In field-based studies, and in particular those studies that have prioritized collection continuously of detailed records of both sedimentological and paleontological metrics of bioturbation, particularly data that allow us to say something about the extent of sedimentary fabric disruption, for instance, through metrics like the ichnofabric index, or those metrics that allow us to say something about the consistency of seafloor sediments close to the ancient sediment-seawater interface, have started to paint a very different picture of early Paleozoic bioturbation than what has historically been envisioned. So for instance, if we look to lower Paleozoic heterolithic siliciclastic strata, so these are thinly interbedded mudstones, siltstones, and sandstones, which is a lithofacies which characterizes a number of different shallow marine and continental margin environments today and in the geologic past. And also because of that lithological differentiation, it offers us a very high resolution look at even small scale changes in bioturbation. So if we look to the lower Paleozoic records of heterolithic siliciclastics, what we see is evidence, again, from abundant and diverse trace fossil assemblages that animals began to colonize these areas of the shallow ocean quite early in their evolutionary history, as early as the Lower Cambrian and even the late Ediacaran. However, the development of extensively mixed sediments, true mixed layer development, was a much more protracted process. And even by the close of the early Paleozoic, some 120 million years after the Precambrian Phanerozoic transition, we can see that the depth of the sedimentary mixed layer, although gradually increasing through this interval, still remains substantially lower than what is characteristic of mixing on the seafloor today. But given what we know about the extent to which bioturbation varies across different regions and different environments in the modern seafloor, can we say anything about paleoenvironmental patterns in ancient bioturbation contemporaneous with these records? And can we even address such fundamental but long-standing questions of whether bioturbation ramped up earlier in more nearshore and more onshore environments or in slightly more offshore settings? So to tackle this question, my colleagues and I have recently begun working in the eastern and southern portions of Newfoundland, Canada. And in particular, we've done some work in the Port-au-Port region of southeastern Newfoundland, in which we have sedimentary strata of Cambrian through Ordovician age, so the same time interval we've been talking about, that are mixed carbonate, siliciclastic in lithology, and that are phenomenally exposed as sea cliffs as well as outcroppings in the intertidal zone. And importantly, this is a succession that not only brackets this critical time period in early animal evolution and the establishment of bioturbation, this is also a succession in which we have a wide range of depositional environments preserved. So allowing us to not only track changes in bioturbation through time, but also to look at time equivalent snapshots from different depositional environments in order to determine environmental patterns in contemporaneous bioturbation. And what we found so far, and some of our preliminary data were published last year in Geobiology, and this is from logging over 350 meters of the Port-au-Port succession and adjacent successions at the centimeter scale, so at the scale at which we know bioturbation varies, is unsurprisingly a fair amount of variability. We do see some strata that are characterized by higher intensities of bioturbation, for instance, ichnofabric index values as high as 5, but these more intensively bioturbated intervals are relatively uncommon. So on the whole, much of what we see in the Port-au-Port succession is very much in keeping with what we saw in those heterolithic siliciclastic strata and other deposits elsewhere, lower to more moderate intensities of bioturbation. And in fact, the mean ichnofabric index throughout the succession is only a little bit over 2. Intriguingly, however, it does seem as though, regardless of their age, those portions of the succession that record our most near shore, our most onshore facies, such as peritidal carbonates and shore face sandstones, are characterized by higher intensities of bioturbation relative to, for instance, the inner shelf portions of our succession or the continental slope facies that we can also examine here. But in order to dig further into why that might be, or whether or not we can actually demonstrate this statistically, we also performed a change point analysis. So in which we allowed a change point algorithm without any sort of a priori binning to determine if or where are the significant inflections in our bioturbation data. So here you're looking at our individual ichnofabric index observations from this succession, arrayed in stratigraphic order from left to right. You can see a little bit of additional information, some relatively coarse paleoenvironmental bins for these units, with the lightest green being the most near shore, darker green being deeper water, and also some lithological bins. And the purple curve here represents the outputs of our change point analysis. And what we can see is that there are a number of significant inflections in ichnofabric index, so in bioturbation intensity, but we don't seem to be looking at any sort of a progressive increase or progressive decrease in this manner. There isn't anything that really resembles a unidirectional trend, just a fair amount of variability. But this corroborates our more anecdotal observations that it is the environments, these strata recording most near-shore environments, such as shore-face sandstones in the Hawk Bay Formation and peritidal carbonates of the Boat Harbor Formation, that are characterized by higher intensities of bioturbation in spite of their great differences in age. So this would suggest that we can in fact infer real environmental patterns in the escalation of bioturbation, that it was our most nearshore environments that at the earliest stage underwent the most tangible increases in bioturbation. However, this does also suggest a note of caution is warranted, and that if we had not collected these data in a facies grounded in an environmental framework, how easy it would have been to potentially mistake environmental variability for an evolutionary trajectory. So thinking a little bit more now about what some of the consequences of early Paleozoic bioturbation may have been. You might recall from my introductory slide, and I know this is something that several of you think about as well, one of the ways in which bioturbators impact their surroundings today is by changing the rheology of seafloor sediments. And they can do this in a variety of ways, but I would say one of the most significant tendencies is that mixing and stirring of sediments can increase their fluid content and porosity and lead to the formation of what we commonly call soft grounds or soup grounds in the upper seafloor. So if we look back in our geologic records, do we see evidence for a similar paleorheological impact of ancient bioturbators? And it turns out that soil marks can offer us just such a proxy. So soil marks are current-mediated deformational and erosional structures. There are a variety of different types, some examples of which you see here, but some of the most commonly employed are tool marks and flute casts, and they're formed either by a tool or by sediment-laden fluids impinging upon and gouging seafloor sediments. And they're best known as paleocurrent indicators because they tend to be very strongly oriented, aligned to directions of flow. However, it turns out they're also excellent paleorheological indicators because a large body of experimental and observational and theoretical work has established that these features can only form in sediments that are cohesive or hydroplastic. They will not form in soupy, well-bioturbated sediments. So if we look back at our geologic archives, can we say anything on the basis of the soil mark record about changes in the erodibility or the rheology of the uppermost portion of the seafloor? And it turns out that if we look at the abundance of shallow marine units containing these features, there's a distinctly non-uniform distribution in time. We see that these sorts of features are considerably more abundant in the early portions of the Paleozoic and the Cambrian and the Ordovician periods. And then we see a tailing off, and this is actually a statistically significant decline in abundance through the rest of the Phanerozoic. And this, of course, very nicely mirrors inversely this interval in which we have independent evidence that the development of sediment mixing of bioturbation was a relatively sluggish, protracted process. And I should note that this trend is robust against a variety of normalization tools. Here I'm showing the abundance of these units normalized to outcrop area. We see the same pattern if we normalize to rock volume or to the duration of our geologic time bends. So this would suggest that ancient bioturbators didn't initially start to really modify the rheology of ancient seafloor sediments during this initial more sluggish ramp up in bioturbation intensity, but presumably eventually they did indeed start to increase the erodibility and the soupiness of uppermost seafloor sediments. But what about the biogeochemical impact of ancient bioturbation? And you might recall I left us with these two apparently dichotomous frameworks. On the one hand, studies that have recently employed geochemical modeling have suggested that the emergence of sediment mixing would have had a negative impact upon both productivity and oxygen levels in contemporaneous ocean waters. And on the other hand, expectations driven by macroecological and macroevolutionary theory might suggest that the emergence of bioturbation, like other forms of ecosystem engineering, should have the opposite effect, should enhance the flow, the trophic transfer of nutrients, and sustain greater ecological complexity. But before we can really dig into whether or not these two frameworks could be reconciled or whether one of them might potentially have shortcomings, I think we need to better understand some of the assumptions that are inherent to each of these. So to start with the first of these, a number of recent studies that have used geochemical modeling to explore the relationship between early Paleozoic bioturbation and contemporaneous phosphorus, carbon, and oxygen cycling have tended to simplify this biogeochemical cycle to essentially a bimodal system, whereby in anoxic settings, it's been assumed that phosphorus delivered to the seafloor with organic matter is preferentially recycled back up to the overlying water column due to anaerobic degradation of organic matter and release of organic bound phosphorus. Whereas in oxic and bioturbated sediments, it's been suggested that phosphorus would be preferentially sequestered and buried in seafloor sediments due to scavenging by iron oxides. And in light of this more bimodal framework, a number of studies have suggested that the emergence of bioturbation would have led to decreases in organic matter burial, increases in phosphorus burial, and that in turn would have starved the oceans of what is ultimately the most limiting nutrient, phosphorus, on geologic timescales, limiting productivity, and decreases in productivity in turn over geologic timescales would lead to decreases in oxygenation. However, this more simplified framework for understanding the phosphorus cycle actually excludes the chief sink through which phosphorus is buried in the oceans today, and that is as authigenic phosphorus that is precipitated in seafloor sediments, particularly carbonate fluoroapatite, or CFA, which is actually the chief phosphorus burial sink, not only along continental margins experiencing upwelling, as we might expect, but also along passive margins, as well as in the deep sea. However, CFA is typically not included in or not fully parameterized in geochemical models, particularly those exploring these deep time feedbacks, because including it does require greater complexity. That properly parameterizing saturation with respect to CFA also requires incorporation of the carbonic acid system, including pH, so it's simply not common practice to include this. Moreover, a number of recent studies that have explored the impact of ancient bioturbation upon contemporaneous biogeochemical cycling using a geochemical modeling lens have tended to simplify bioturbation to a single term. However, as of course all of you are very much aware, bioturbation is not a single process. It's an umbrella that encompasses a wide range of behaviors through which animals interact with sediments. Some of these behaviors we can loosely group together under the term biodiffusion, or the randomized movement of sedimentary particles, so essentially sediment mixing and mixed layer development. And in geochemical models, we can represent biodiffusion with an intensity coefficient, dB, as well as with an attenuation coefficient, so essentially a depth, xbt. And others of these animal sediment interactions we can loosely group together under the term bioirrigation, which is the non-local movement of pore water solutes, for instance, open burrows, such as this burrow was once upon a time before it became infilled with sand. And as we do for biodiffusion, we can represent bioirrigation with an intensity coefficient alpha, as well as with an attenuation coefficient xbi. And although it's been common practice in exploring the relationship between ancient biogeochemical cycles like the phosphorus cycle and ancient bioturbation to simplify each of these aspects, I would argue that if we really want to understand the mechanisms that underpin these interactions, we need to have more realistic representation of each of these processes. So toward that end, my colleagues and I a few years ago developed a new 1D diagenetic, so seafloor geochemical, or commonly called reactive transport model, which we called SEDChem. And SEDChem, like all diagenetic models, is built upon Bob Berner's simple diagenetic equations, which you can see here, in which changes in the concentration of both dissolved species and solid phase chemical species over time and with depth in the sediment pile are a function of not only a range of different chemical reactions, but also transport processes such as diffusion or the advection associated with sedimentation, or for that matter, bioturbation. And SEDChem includes over 40 such reactions, including a full suite of microbially mediated redox reactions, as well as carbonate chemistry, including CFA formation, and multiple styles of bioturbation, both bioirrigation and biodiffusion. In using SEDChem, we can closely replicate empirical, real-world, both pore water and solid phase geochemical data from a variety of marine settings around the world. So what you're looking at here is the correspondence between our SEDChem model outputs, which are the orange curves, and with all these symbols, empirical observations from the particularly well-characterized Friends of Anoxic Mud or foam site in Long Island Sound, which is here on my back doorstep in Connecticut. And foam is a site that is perhaps unique in the extent to which it has been characterized in detail, both from a geochemical perspective, including phosphorus speciation analyses, as well as from the perspective of bioturbation. And we can see that we have a fairly close correspondence between our model outputs and our empirical observations, and that's without need for any additional parameterization or tuning. But I also want to stress that SEDChem's performance is not contingent upon boundary conditions or a reliance upon conditions that we see at foam. We've also repeated the same exercise for other settings around the world, including the deep sea. And subsequently, having ground truthed our model, we've used SEDChem to explore the relationship between different intensities and styles of bioturbation and biogeochemical cycles, such as the phosphorus cycle. So what you're looking at here on this slide are the outputs of a series of different sensitivity analyses in which I have modulated each one of those 4 bioturbation parameters I introduced previously while holding all other factors constant. And I've organized this with the 2 biodiffusion parameters, our biodiffusion intensity coefficient, dB, as well as our biodiffusion attenuation coefficient, or depth, XBT, over here on the left, and our 2 bioirrigation parameters, including our bioirrigation intensity coefficient, alpha, and our bioirrigation attenuation, XBI, on the right. And you can see that these are represented fractionally along the x-axis. So 1 means 100%, which corresponds to the values that we observe at that foam site. But you can see the full range of values we explored listed below. And then along the x-axis, you can see a couple of different representations of phosphorus cycling. The solid curves correspond to CFA, so our major phosphorus burial sink, authigenic phosphorus. And the dashed lines correspond to total phosphorus burial efficiency, or the balance between inputs of phosphorus to the system and outputs in our variety of burial terms, including CFA, but also organic bound phosphorus and iron oxide sorbed phosphorus. And I'm showing you each of these so that you can see that changes in total phosphorus burial efficiency do in fact closely follow and are driven by changes in CFA. And lastly, we can see a couple of different colors here. The green curves correspond to higher maximum XBI values, so essentially greater maximum depths of bioirrigation, and the blue curves correspond to more moderately bioirrigated sediments. So stepping back to a first order, we can see that bioturbation has a complex, frequently non-linear, and in cases non-unidirectional relationship to phosphorus burial. And bioturbation and biodiffusion and bioirrigation actually appear to have competing effects upon phosphorus burial, which is something that previous studies hadn't noted. So increases in biodiffusion can lead to increases in phosphorus burial, whereas increases in bioirrigation appear to lead to decreases in phosphorus burial. And if we dig into this a little bit more mechanistically, this actually makes a good deal of sense. Biodiffusive sediment mixing, by bringing sedimentary particles, both organic particles and phosphorus particles, deeper into seafloor sediments, brings them into what are commonly more anaerobic regions of the sediment pile, where under higher pore water pH values, it can be easier to achieve saturation with CFA. But also at those greater depths, any phosphate liberated from those phases, it's going to have a much more challenging time effluxing back up to overlying ocean waters where it can escape. In contrast, although bioirrigation can increase the possibility of iron oxide mediated scavenging of phosphate, it also increases the relative importance of aerobic degradation and lower pore water pH values, so higher, more challenging to achieve saturation with CFA. And it's also simply much easier for phosphate liberated through those pathways to then simply escape back up to overlying ocean waters, where it will be recycled but will not end up buried in the sedimentary record. But if we think back to what we've learned from our stratigraphic archives and recall that these suggest we have abundant colonization of the seafloor, abundant burrow constructions quite early in the evolutionary history of animals, but that sediment mixing is not keeping pace with that. That might suggest that in many early Paleozoic shallow marine settings, bioturbation was dominated by bioirrigation associated with burrow construction, with domicile construction in particular, and therefore increases in bioturbation through this interval may have led to decreases in phosphorus burial and enhanced phosphorus recycling, in contrast to the strictly positive relationship between bioturbation and phosphorus burial that previous modeling studies had suggested. But bringing us more in line with the expectations of macroecological and macroevolutionary theory, that the ramp up of bioturbation, at least initially, may have led to greater phosphorus recycling, fueling greater productivity. Intriguingly, however, if we consider our more deeply bioirrigated systems, so here looking at the green curves, subsequent increases in biodiffusion, such as we would infer were eventually the case, don't actually appear to substantially impact phosphorus burial. And in fact, phosphorus burial efficiency in these systems is indistinguishable from that of modern sediments without any bioturbation at all. So if we start to summarize some of what we've learned from this coupled record-based and geochemical modeling exploration of the evolution of bioturbation, I think that we have abundant evidence from both sedimentary and paleontological archives that the development of intensive sediment mixing was a very protracted process. And I think we should stop and consider the scale of this lag between the early stages of animal colonization of the seafloor, as beautifully preserved by the trace fossil record, and the intensification of sediment mixing and the development of a deep mixed layer. This is a really striking macroevolutionary lag that spanned over 100 million years, and including a number of important radiation events in early animals. So why might it be that we have such a striking macroevolutionary lag? And I think one possibility that we should at least consider is whether this could reflect a delay in the emergence of the necessary developmental or physiological toolkit. So were early bioturbators simply incapable of more intensive forms of sediment mixing? And although I think this is a fine null hypothesis, I think that this idea has less credence because although rare, we do see intervals characterized by higher intensities of bioturbation. For instance, ichnofabric index values as high as 5, suggesting that animals didn't lack the ability to intensively bioturbate, but some other factor, either ecological or environmental, kept the extent to which intensive bioturbation was expressed somewhat in check through this interval. And I think one intriguing theory lies in some of the work of Charles Thayer, who in the late 70s and early 80s suggested that the interval immediately following what we've been focused on here, the middle and later Paleozoic, was a time of major radiations and what he called the biological bulldozers. So our mobile deposit feeding animals that today include a number of different types of bioturbators, from holothurians to echinoids to a number of crustacean groups, and which are particularly impactful bioturbators because in many cases they're large, they're also continually on the move, and they're deposit feeding. So not only physically, but they're also chemically processing sediments as they move. And Thayer based this surmise of a middle to later Paleozoic radiation of the bulldozers upon the body fossil record of another group of organisms, those organisms that are immobile and that are suspension feeding and live in muddy substrates. And he observed pretty striking declines in the abundance of these muddy substrate suspension feeding taxa during this same interval. And presumably these would have been the organisms that were most susceptible to the disturbance of seafloor sediments, as well as the increasing turbidity of the benthic boundary layer that may have accompanied the radiation of biological bulldozers. But this is a hypothesis that one of my PhD students, Kate Pippinger, is currently working on testing by collecting primary records of sediment mixing for the Middle and later Paleozoic. However, I think that we shouldn't discount the possibility that environmental factors could have also played a strong role in shaping this delay. In fact, there is a large and growing body of geochemical evidence suggestive that the low seawater oxygen concentrations characteristic of the late Precambrian may have persisted well into the Paleozoic. Bioturbation is, of course, a fairly metabolically demanding behavior. So if we couple low oxygen concentrations with the very high temperatures that have been reconstructed for seawater for portions of this interval, it may be that those environmental factors serve to keep intensities of bioturbation in check, apart from those settings that are most shallow and most wave reworked, and thus going to be the best aerated. And of course, there are also substrate factors. If seafloor sediments were fairly firm and cohesive through portions of the early Paleozoic, as our Solmark record has suggested, then it may have been particularly challenging for bioturbators to move into and turn and mix up those sediments. So perhaps the radiation of the biological bulldozers was important not only in its own right, but also by serving as a form of icebreaker and making upper seafloor sediments more accessible to smaller and less efficient bioturbating taxa. But if we return to some of what we've learned from these geochemical modeling exercises that I've shared with you, if early Paleozoic bioturbation was dominated by bioirrigation, as potentially suggested by that trace fossil record and by the paucity of well-mixed sediments over this interval, that might suggest that bioturbation increases through this time would have served to increase phosphorus recycling rather than increasing phosphorus burial, as previous studies had suggested. But allowing us to reconcile those two historically contradictory frameworks and suggesting that bioturbators may indeed have served to foster gradient nutrient exchange and ecological complexity more along the lines of the expectations of macroecological and macroevolutionary theory. And finally, I think that if we're trying to reconstruct the interactions between bioturbation and biogeochemical cycling, especially in this critical interval of early animal history, if we not only need more robust first order records, allowing us to reconstruct changes in bioturbation through this time and across different environments, but we need to examine these interactions, the net effect and even the directionality of which may not be intuitive in a more mechanistic modeling framework. And then for just very briefly for the last a couple minutes, I wanted to share some of the recent work that my group has been embarking on over the last year or so. And some of that has entailed thinking about how these sorts of feedbacks, for instance, feedbacks between phosphorus cycling and bioturbation, may have shifted during different climate states, and in particular, how they would have been affected by warming, whether past hyperthermal episodes or current and future projected warming associated with anthropogenic emissions. So this is a study that we published last year in American Journal of Science, in which we used the same diagenetic model, SEDChem, to explore what warming may have meant for bioturbators, and in particular leveraging published data sets such as Ouellette et al. That allow us to try to trace out some of these parabolic response curves that all organisms should have in response to temperature, but which for bioturbating taxa, and in particular their levels of intensities is notably under constrained. So thinking about how organismal bioturbation activity will shift under different temperature regimes, but also coupling that with a more quantitative exploration of the number of other biogeochemical factors that may shift with warming, such as rates of microbial respiration, of sulfate reduction, or rates of autogenic mineral precipitation, such as CFA precipitation, or the temperature effects upon the solubility of oxygen in ocean water, or even changes in continental weathering and the supply of fresh phosphorus to the ocean system. And the um, the summary is that it's really not simple, as you might expect, given the number of feedbacks involved, and it really depends upon the time scales that you examine, whether immediate organismal scale responses or longer term responses linked to weathering and to carbon burial and associated feedbacks in the phosphorus, carbon, and oxygen cycle. But I think one of the things that did emerge from this work is how important that it is that we continue to build empirical data sets constraining how bioturbators and their level of activity will shift under warming. And this is an area that we've recently undertaken exploring in more detail in my lab with a variety of incubation experiments looking at different bioturbating taxa and their level of activity under different temperature and oxygen regimes. And lastly, one other avenue of bioturbation carbon cycling that we've been exploring, again through a diagenetic modeling lens, is thinking about blue carbon ecosystems, which are, of course, incredibly important from an ecological perspective, but also have long been in the public view for the potential benefits that they provide to offsetting carbon emissions through sequestering organic carbon. More recently, however, there's been a great deal of interest in these systems and systems like mangroves and seagrasses from the perspective of more durable forms of carbon storage, for instance, the manner in which they can enhance alkalinity. And by dissolving carbonate sediments through increased respiration of the root structures of these organisms and increased sulfide oxidation of organic matter, the manner in which that can foster carbonate dissolution, alkalinity enhancement, and thus, through a re-equilibration of the carbonic acid system, foster greater drawdown of atmospheric carbon dioxide into the shallow oceans. And this is work stemming from a study published last year by Fakhri et al. In Nature Sustainability. So what we've been doing over the past few months, working with Moji Fakhri and others at Yale, as well as Tom Bianchi at University of Florida, is implementing a bioturbation perspective into this same diagenetic model. And in particular, thinking about the impacts of warming on keystone bioturbators like Uca, Pagilitur, the Atlantic sand fiddler crab, which is of course an important bioturbating species in a number of systems, including tropical mangrove environments, and what the impact of warming and thus shifts in the level of activity of organisms like Uca might mean for carbon storage potential in these systems. So here you're looking at a variety of outputs from our diagenetic model simulations. We have a baseline scenario, which is basically business as usual, current temperatures, current levels of biodiffusion. Then scenarios 1 and 2 represent progressive increases in warming and projected increases in biodiffusion intensity. So again, thinking about the rising limb of that parabolic response curve. Initially, bioturbators may be receptive to higher temperature levels, and may compensate by increasing their levels of bioturbation. However, with scenario 3, we're mimicking the falling limb of that metabolic response curve, and thus seeing a major decrease in, perhaps even mortality, of bioturbators like Uca. So what do we see? So we see that as we increase levels of biodiffusion and increase temperature, including temperature controls upon rates of organic carbon remineralization, is that bioturbation initially increases, and this can lead to increases in the oxygen penetration depth, but at the same time increases in organic carbon burial efficiency by shuttling more organic carbon away from superficial sediments into the deeper sea floor. And that has the effect of making less organic carbon available for respiration, but also increasing the importance of aerobic respiration pathways, which results in a shift to more carbonate undersaturated conditions and less alkalinity generation. Whereas when we knock out our biodiffusers, then we see a corresponding decrease in oxygen penetration depth and organic carbon burial efficiency, and a rebound in alkalinity generation. So the upshot is that the impact of bioturbators or their responses to warming can potentially, at least initially, mitigate the extent to which blue carbon ecosystems like mangrove may serve to foster more durable forms of carbon storage. And thus, I think that it's critical that when we think about natural solutions and ecological as well as carbon cycle solutions to ongoing anthropogenic emissions, that we factor in the role of bioturbators and other ecosystem engineers in modulating the potential of these ecosystems. And with that, I'd like to thank a number of folks who were integral to this work, as well as various sources of funding, and all of you for your attention. I'm happy to take any questions.
That was a great talk. I can start with a question while other people are waiting. So then this is kind of like a broader question, but I think it's really interesting that, you know, there's this ichnofabric index that's really based on kind of the level of physical mixing. And what we do in modern times is mostly quantified dB, which is essentially a chemical tracer. And, you know, I mean, it's a very different metric. And I'm wondering, you know, like one of the big goals with this is to like bring our modern methods together with the paleo methods. So like, what can we do studies that would better course, you know, like we can do x-rays and look at like, you know, stratigraphy of x-rays, or but like what, how do you merge those really different methods?
Yeah, that's a great question. And I think I would pitch it maybe almost the opposite, that in deep time, we don't have anywhere near the level of resolution or the sort of methodological toolkit that those of you who work in the modern have available to characterize these things. So I think we need to sort of continue to think creatively about what we can say in a more quantitative or even semi-quantitative manner about intensities of bioturbation in the past from the rock record. Like so many of the metrics that folks use widely in the modern, like radio tracers, or other particulate tracers or fluid tracers, are simply not accessible to us in deep time. And of course, in general, sort of backing out correspondences between bioturbation and biogeochemistry in deep time can be really challenging, because for rocks of this age, we don't have drill cores from the seafloor, right? This is beyond the age of our oldest seafloor sediments. And sometimes the best places to go and look at these trends are places like uplifted terrains in desert regions, which are, of course, like the worst place you can go to try to back out most of the geochemical proxies that folks are interested in sort of comparing to bioturbation. So that sort of poses another set of challenges, and one that there's not necessarily a straightforward way to overcome. I would say the sort of proxies that my group uses from the sort of like the ichnofabric index perspective are sort of most comparable to things like X-ray analyses that folks use in shallower time and in the modern. So there's that sort of connecting thread. But another question that I've been interested in of late is like how much offset is there between the different modern proxies? Like when we're trying to compare deep time bioturbation to modern bioturbation, do we need to limit ourselves to a particular proxy? Can we look at the sort of cumulative data set that's informed by many proxies? So I think there are still like a number of important questions to resolve on both sides, both the modern and the ancient, to better be able to connect those two very rich data sets.
Yeah, it's a big challenge. It's interesting. And then because there's the time, and then there's the chemistry versus the physics, and then you know, there's just a lot of go ahead, Bob.
Sorry, I joined late, so I apologize for that. I'm going to have to go back, and hopefully we have access to recordings. Is that right, Kelly?
Yeah, Adrian's recording.
I'll have to look at your earlier presentation, but I just wanted to ask about the depth of bioturbation that you've fixed on for a lot of the modeling and for your statements about blue carbon storage and so forth, like in mangroves. And my experience in mangroves is that the Euca and Eucites that are dominant in those systems, they often go deeper than a meter in terms of their burrow galleries, and their interactions with mangroves are actually extremely important over those larger depths. So I'm wondering if the utilization of the Boudreau mixed depth, which is a lead-210 based mixed depth of 10 centimeters, plus or minus 10 centimeters, is misleading in terms of the impact of bioirrigation on these systems, in the sense that it makes us zero in on a 10 centimeter scale length, which in fact, the depth of impact can be far greater. And even in Long Island Sound, which I just caught the last part of what you were talking about, the foam site, I know that the squilla, the mantis shrimp that's present throughout Long Island Sound, they go down to 3 meters during the winter in Long Island Sound. And this has a huge impact on the, not so much the dB that you would derive based on a typical core size, but the irrigation and exchange of the pore water down to several meters depth. So can you comment on the record of when in the geologic we really get these much deeper systems formed?
Yeah, absolutely. And of course, a lot of that work in Long Island Sound was very much informed by your work. So first of all, absolutely. Like, I think, one of the big outcomes for me of this modeling exercise, even, sort of the more facile sensitivity analyses that were our initial focus, is driving home, the same point that you just made, that bioirrigation and biodiffusion are not the same thing, and they can have very different impacts upon biogeochemical cycles. And that's also something that I think that, more of the sort of broader community, both the paleontological community and the folks who are doing sort of biogeochemical modeling, say in the Mesozoic and the Cenozoic, that's a really important message, that burrow depth and mixed layer depth are not the same, and burrow depth can greatly exceed the depth of the sedimentary mixed layer, and also that the biogeochemical consequences of each of those may be, in some cases, quite different. So I entirely agree with you there. In terms of the record, that's actually a really intriguing question, and something that we've sort of been working on in parallel to this sort of mixed layer investigation that I talked about today. And in the interest of time, did not have time to talk about some of the work we've been doing on sort of reconstructing what is our sort of state of the art. What do we know currently about the evolution of the transition layer through time, as approximated by our deepest burrows. And that's something that my student Kate that I alluded to, who's been working on middle and later Paleozoic mixing, has also been very much involved in, and this is a collaboration with folks like Matt Clapham and Dave Botcher, as well as Mary Drozer and others, and Chuck Savda. And what we found so far is that if we sort of perform a database centric approach to compiling all the burrow depth information, especially for deeper burrowing taxa like trace fossil genera like Scolithos and Arenicolaides and Diplocrateri and Zoophycus, Ophiomorpha, for instance, is that that gets started really early on, largely through things like Scolithos pipe rock, which has long been known as sort of a phenomenon of earliest Paleozoic shore-face sandstones, is that we have these really deep cylindrical burrows that get established really quickly. There are other significant inflections later on that we think might be associated with the Mesozoic marine revolution, where we see our U-shaped burrows and some of our other ichnotaxa substantially and significantly increase in burrow depth. So there are later changes as well, but on the whole, deep burrowing gets established really early on. in contrast to what we see with intensities and depths of sediment mixing. In terms of the biogeochemical consequences of that, that's another fascinating question. And for schlitzose pipe rock in particular, that's something that we've started to dip a toe into. One of my recent postdocs, Sophie Westicott and Mingyu Zhao, and myself have recently embarked upon trying to interrogate through a lens of diagenetic modeling. And so far we see that the impact of scalithose pipe rock, although measurable, is actually relatively minimal compared to other factors that influence, for instance, extensive sulfide oxidation or the balance between aerobic and anaerobic forms of carbon respiration. So it's a fascinating question. I think it's exactly sort of on point. These are the questions that we need to be asking, but there's been sort of limited quantitative forays in that direction for deep time, and I hope that's something that's going to continue changing in future.
Great, thank you. I'm having all sorts of computer problems, but I did hear your reply. So thank you so much. I'm going
No
to have
worries.
I'm going to have to access much of this, as I said, on recording, and I'm sorry about that. But
no problem. Thanks for joining, Bob.
yeah,
And thanks for the great question.
Does anybody else have any more questions? We're right at 11 o'clock, so we're kind of at our hour limit. But if there's another question, we could do that. I have a quick one, if that's okay.
Yeah, please.
People can take off if you want. If I can ask one. So I was interested in the soul mark stuff you were talking about, and the fact that you don't see those in sands and you don't see them in really goopy muds, and so I've been thinking a lot about small scale geotechnical properties and how that affects sort of this ecosystem engineering. And I'm wondering how much people have looked at like the paleo. Can you look at fossils and what can you pull out about the geotechnical properties? Like, I assume you can get like grain size and distribution, but have people really thought about, what does that mean in terms of the plasticity and stiffness and cohesion and any of those properties?
Yeah, no, that's a great question. So from like a more geotechnical side related to like porosity and permeability, that is something that folks have investigated, especially those folks who have played a big role in sort of developing the data set that have informed the energy industry, for instance, and thinking about hydrocarbon plays and where they might be located. So that is information that at least where like the further back you go in time, and of course things that have sort of undergone more diagenetic and metamorphic processes, you can get sort of orthogenesis can make it harder to, like cementation can make it harder to sort of more simply measure things like grain size. But in general, that is something that if there hasn't been too much recrystallization, we can do. I would say sort of other sort of paleorheological indicators. I think that from a bioturbation perspective, that soul mark study is one of a regrettably small number, but there are some folks who have thought about this for quite a while. Like actually this fell along some of the work that folks like Don Rhodes and Josie Aller did, even decades ago, really pioneering work looking at changes in sediment cohesiveness related to bioturbation, as well as to other physical and ecological processes like microbial colonies, or Don's work looking at changes in porosity associated with suspension feeding and deposit feeding communities in places like Buzzards Bay, also played a big role in sort of influencing me to sort of try to find new ways to tackle these questions for deeper in time. So the soul marks are, one way that we can sort of try to gauge that is, what were the sort of cohesive properties of muds. It's not particularly quantitative, but I think from a sort of descriptive level, it can be very powerful in telling us about sediment consistency in the upper portion of the seafloor. One of the other things we can look to is sort of the fidelity of preservation of those trace fossils themselves, particularly if we have reason to believe that they formed very close to the paleo sediment water interface. And thus are telling you something about substrate conditions close to that sediment water interface. So features like bioglyphs, which de Chabert and Eckdale described, a decade ago, of things like scratch marks and very delicate ornamentation of burrow walls, or even the sort of podite scrapings of many of our arthropod trace fossils, like the one on my title slide, the Rusophycus that you saw there. Those similarly can really only form in very cohesive and hydroplastic sediments. So I think the trace fossils themselves, and when they're exceptionally preserved, which I think as an ichnological community, sometimes we take for granted, when that happens, we are very appreciative of the way it opens windows into inferring something about the organism's physiology and its behavior, but we don't always stop and think, what led to that extraordinary preservation? Like, what is it that enabled, scrapings of somebody's claws to actually become preserved in that manner? To how did they form, and how did they actually end up in, hundreds of million-year-old rock records? So I think those can be enormously informative as well in telling us about sort of the geotechnical properties and the cohesiveness of those shallow seafloor sediments.
Well, a lot of interesting questions. So we should probably wrap up unless anybody has any final questions. And then Adrian will post the video at some point. But yeah, this was a great talk. Thank you so much for joining us.
Well, thanks again for the invitation to join. It was a lot of fun, and I'm looking forward to the upcoming talks in the seminar.